Do you remember what I said about the pyruvate dehydrogenase complex from earlier? Well, the pyruvate dehydrogenase complex is going to link glycolysis, which occurs in the cytoplasm, to our TCA cycle, which occurs in the mitochondria. The net reaction is going to convert a pyruvate, NAD, and coenzyme A into three products, an acetyl-CoA, CO2, and NADH. Here, we can see pyruvate dehydrogenase creating an activated carrier, an NADH molecule, when it converts a pyruvate into an acetyl-CoA. Now, there are going to be five cofactors that this pyruvate dehydrogenase complex needs to function properly. We'll need a pyrophosphate, which is basically two phosphate groups, and this is supplied by vitamin B1 in the form of a thiamine pyrophosphate. We'll also need FAD, NAD, and CoA, which are going to come from vitamins B2, B3, and B5 respectively. On a side note, the last one, lipoic acid, is going to be inhibited by arsenic, and arsenic poisoning will cause vomiting, rice water stools, and garlic breath. If you're exercising, you'll probably want to be undergoing some sort of aerobic metabolism, and therefore you'll want to keep the pyruvate dehydrogenase complex active. The body's main goal will be to link glycolysis to the Krebs cycle, the TCA cycle, using this pyruvate dehydrogenase complex. So during exercise, the ratio of NAD to NADH is going to increase since you're consuming all of that NAD during glycolysis to make a more reduced form, that NADH, which will then go on to the mitochondria and exchange it to make lots of ATP during oxidative phosphorylation. Other changes during exercise include an increase in the ADP levels, because you're burning up all your ATP, and also an increase in calcium levels, which are then going to go on to activate your PDH complex. Pyruvate dehydrogenase is going to be inhibited by stuff such as ATP, which makes sense. If ADP activates it, ATP is going to inhibit it. But also you'll have high NADH levels, and then also acetyl-CoA levels. So all of these are going to be found downstream from the PDH enzyme, so we're going to see some negative feedback by these products. Where can we see a similar enzyme to our pyruvate dehydrogenase complex? Well, remember, in the TCA cycle, you're going to see the alpha ketoglutarate dehydrogenase enzyme. This complex is going to convert alpha ketoglutarate to a succinyl coenzyme A in our TCA cycle. And this is going to use the exact same set of cofactors as our pyruvate dehydrogenase complex. So if you can remember one of these, you can remember the other as well, since they're both going to use the same set of cofactors and that both are dehydrogenase complexes, which are going to decarboxylate their substrates. Let's talk about a clinical application of the pyruvate dehydrogenase enzyme. A pyruvate dehydrogenase complex deficiency is a serious problem for all cells and all mitochondria since it prevents the products of glycolysis from actually entering the TCA cycle. And therefore, it's actually going to prevent aerobic metabolism. The pyruvate from our glycolysis normally gets converted into an acetyl-CoA molecule, and in the process, we get our activated carrier, the NADH. However, if this pyruvate cannot get converted into an acetyl-CoA molecule because of a pyruvate dehydrogenase deficiency, then it'll get shunted into anaerobic metabolism, and it gets converted into a lactate molecule. This pyruvate dehydrogenase deficiency can be either congenital, meaning they're born without this enzyme, or a defective copy of this enzyme, or it could be acquired later on in life. Now, why do you suspect alcoholics can present with an acquired form of pyruvate dehydrogenase complex deficiency? Well, this is because alcoholics typically are nutrient deficient, especially in vitamin B1. Thiamine, which remember I said was listed as a cofactor for the pyruvate dehydrogenase complex. Boards love testing this concept, so don't be surprised if you see this on test day. Clinically, a pyruvate dehydrogenase deficiency is going to manifest as neurologic deficits, or a lactic acidosis because of a high level of lactate, or as an increase in alanine in serum during infancy. It's also treated by increasing the intake of ketogenic nutrients, such as a high-fat diet or the high diet of ketogenic amino acids. lysine, and leucine. Well, that's strange. Why a diet high in leucine and lysine? Well, these nutrients, the leucine and lysine, are going to not go through the pyruvate dehydrogenase complex to enter in the TCA cycle. So here we see our glucose gets converted to pyruvate via the process of glycolysis. Our pyruvate normally gets converted into an acetyl-CoA via this pyruvate dehydrogenase enzyme. And our acetyl-CoA is going to go into our TCA cycle. However, our leucine and our lysine are ketogenic amino acids, meaning they're going to bypass the PDH complex and thus get fed into our system at a further downstream location. So it's actually going to bypass the PDH complex. And then also notice how pyruvate can also be converted via anaerobic metabolism into our lactate, which I said earlier. Let's do a quick recap. We spent all that time talking about glycolysis, which the endpoint of glycolysis, remember, was to make two molecules of pyruvate from one molecule of glucose. Pyruvate is an important intersection point for several metabolic pathways. So what are the different pathways a single pyruvate can take? Well, first, it can be converted to alanine in the liver via alanine transaminase, or ALT enzyme. Now, this is how amino groups are transported from the muscle to the liver. Secondly, pyruvate can be converted to oxaloacetate via pyruvate carboxylase to either replenish the TCA cycle or be used for gluconeogenesis. Third, on our last slide, we just said that pyruvate can link glycolysis to the TCA cycle using the enzyme pyruvate dehydrogenase complex. So we've already seen that one. Then fourth, pyruvate itself can basically be the endpoint for energy production in anaerobic glycolysis, which we've already kind of covered. It can be converted to lactate via the enzyme lactate dehydrogenase. Now, this reaction is going to regenerate our NAD+, so that glycolysis can actually continue to produce small measly amounts of ATP. Where in the body do we typically see our anaerobic metabolism? Anaerobic metabolism is the main source of ATP for red blood cells, leukocytes, or white blood cells. It's seen in the renal medulla, it's seen in the lens of the eye, and then also the testes and the cornea. These are all sites of anaerobic metabolism, where they lack the aerobic metabolism. Let's now go into detail about the TCA cycle. Thinking back to our discussion on metabolism sites, do you remember where the Krebs cycle takes place within a cell? Well, the TCA cycle is going to take place in the mitochondria of a cell, and it's going to produce the high-energy electron carriers NADH and FADH2, which are going to be used to generate ATP in the electron transport chain. This is going to complete our metabolism of glucose. This diagram shows the basic steps of the TCA cycle. Notice how acetyl was fed into the system, and the cycle spins, which creates various activated carriers, which we talked about earlier. It also produces a molecule of GTP, which is the equivalent of an ATP molecule. And once the cycle is done, the TCA cycle is ready for another input of acetyl-CoA, and it'll keep on rolling. This is a very basic example of how the TCA cycle looks. We'll now add in some enzymes and intermediates to this cycle. Pyruvate is going to be the main substrate. So pyruvate was created from our glycolysis, and then we created one molecule of glucose, created two molecules of pyruvate, and that pyruvate is going to get converted into acetyl-CoA through pyruvate dehydrogenase complex. And in the process, we're going to get out an activated carrier, an NADH, and also a CO2 in the process. So this is kind of how we got to where we are. So now we have a molecule of acetyl-CoA. Now, also as a side note, I want to find out that we can get proteins and lipids that feed in at this point in the cycle. Notice how we had carbohydrate metabolism through our glucose, protein metabolism, and also fat metabolism. These are all connected at this step in the cycle. So let's go ahead and clear that off. The net reaction for the Krebs cycle is going to be to produce 3 molecules of NADH, 1 molecule of FADH2, two CO2s, and a GTP, which is an ATP equivalent. So this all amounts to 12 ATP per acetyl-CoA molecule entered. Where do all of these activated carriers end up ultimately? Well, these activated carriers can then be exchanged later on in the electron transport chain to produce ATP molecules. We'll see later on how each NADH and each FADH2 is fed into the system. NADH can be exchanged for 2.5 ATP per NADH molecule, and each FADH2 can be exchanged for 1.5 ATP. So, since glucose yields two molecules of acetyl-CoA, so one molecule of glucose yields two molecules of acetyl-CoA, we can pretty much multiply this 12 by 2, so we'll actually get 24 molecules of ATP from each glucose molecule. The TCA cycle is regulated at three major irreversible steps, which are catalyzed by the enzyme citrate synthase, isocitrate dehydrogenase, and also alpha-ketoglutarate dehydrogenase. By the way, as a side note, you don't have to know anything about the actual structures for these molecules on test day. The names will suffice. Okay, so let's take a look at our first enzyme, citrate synthase. This can be inhibited by high amounts of ATP in the body. Because remember, if you have high amounts of ATP, well, the Krebs cycle is used to make ATP. So if you have a lot of ATP, you don't need to run this Krebs cycle as much. So we're going to take that ATP, inhibit the very first enzyme in this cycle, thus we're going to prevent the Krebs cycle from producing even more since we have plenty of ATP. The citrate synthase enzyme is going to combine an acetyl-CoA with an oxaloacetate. And notice how oxaloacetate is kind of the end product of this cycle, so we can keep regenerating this cycle. Now citrate synthase is going to synthesize a citrate molecule, citrate synthase. Okay, our second enzyme, isocitrate dehydrogenase, is going to convert an isocitrate into an alpha-ketoglutarate. And in the process, we're going to produce an NADH molecule. Notice also that we lost a carbon as a carbon dioxide, and it's also logical that this enzyme is going to be inhibited by high amounts of ATP, which we just talked about, but also it's going to be inhibited by high amounts of NADH because we're making an NADH in the process. If we have plenty, we don't need to run this cycle. Our last irreversible step is going to be the alpha-ketoglutarate dehydrogenase enzyme, which converts an alpha-ketoglutarate, which we just made, into a succinyl-CoA. As I mentioned before, the alpha-ketoglutarate dehydrogenase complex uses the same cofactors as the pyruvate dehydrogenase complex. Remember this pyruvate? And then we had our pyruvate dehydrogenase complex. So we're kind of seeing a bigger picture here. They use the same cofactors. Do you remember what those cofactors were? Well, our pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase complexes are going to use vitamins B1, vitamins B2, vitamins B3, B5, and then also lipoic acid. So 5 cofactors there. And as a side note, do you know which important synthesis pathway starts with a succinyl-CoA molecule? What pathway uses a succinyl-CoA enzyme as the starter? Well, that would be heme synthesis. So this is an important concept. I'm just trying to illustrate that this cycle has many different important factors. The TCA cycle is responsible for creating ATP, but it also creates intermediates that can be fed into other pathways within the body. Let's take a look at a flash quiz. At what steps in the Krebs cycle are NADH molecules, GTP molecules, and FADH2 molecules produced? Well, the question is asking where our activated carriers are being created. NADH is going to be made at 3 different points in the body. The conversion of isocitrate to alpha-ketoglutarate, alpha-ketoglutarate to succinyl-CoA, then also malate to oxaloacetate. GTP, an equivalent of ATP, is going to be created when succinyl-CoA is converted into a succinate molecule. Then lastly, our FADH2 molecule is going to be created when we convert succinate into fumarate.